So thanks everybody for coming to our first ever Drug Hunter webinar. I am so excited to finally be doing some of these things sort of live. I wish I could see your faces and say hi to people, but we'll have to save that for later. I will be at AAPS in Boston hosting a symposium on preclinical compounds moving from preclinical to clinical development. And we are still looking for 2 speakers for that session in October. So if you know anybody good that you want to give a talk in Boston, please let me know. And we will also be hosting a live event in Boston as well. So if you're interested in that, stay tuned and get in touch with Melissa. Okay, so for this first group of webinars, I'll just be doing a review of things from the literature, the news, events, things like that. We do plan to have guests on, and I'm very excited about a couple people that have accepted our invitations to speak. But just like any other seminar series, if you guys have any people you want to invite as an invited speaker, please let me know, and we will make it happen. All right. So in this section, we're going to just run through a couple highlights from approvals of Q1 2022, some of the molecules of the month that we found were cool for a diverse range of reasons, and some conference disclosures from ACS and AACR. Now, I had originally wanted to cover emerging startups, growing companies, clinical readouts, positive and negative, acquisitions and partnerships, etc. We have a lot of research there that I can't wait to show you, but as I was preparing this talk, that would have been way too huge. So we're going to break this up into 2 sessions, and you're automatically signed up for the second session, or if you haven't signed up for the series, watch out for that one as well. So that'll be coming in a week or 2. Just to give you a preview of what we'll be talking about there, lots of new innovative companies with interesting new approaches or pipeline molecules. We'll just be breezing through those as well as other events and clinical readouts that people should pay attention to. All right, so for approvals, there were 10 new molecular entities approved in the first quarter. We're keeping a live tally on the site, so you can search for 2022 drug approvals and you can find a page that groups all of them together. I'm not going to highlight all of them, even though they were all interesting, but I'm just going to pick out some snippets from each story. And the first one is Q-Vivic. I feel like they make these harder to pronounce each year. Starting with a Q is an interesting move. Daridorexant is a dual OX1, OX2 blocker. So orexin is a peptide that promotes wakefulness. And so if you can block the interaction with its receptor, it can help promote sleep. So this drug is for insomnia. What I thought was interesting about it was typically when we think of making a best-in-class molecule, we maybe think of longer half-life, lower dose, smaller pill, things like that. The differentiation strategy here, so this is the 3rd orexin antagonist to be approved. It actually has a shorter half-life of 8 hours than suvorexant and lemborexant. And for insomnia patients, this is important because this helps reduce morning grogginess after your insomnia medication's done its job and you've woken up and are going to work. You don't need it to be working anymore, especially if you're taking it for a couple days. Another interesting tidbit is this molecule is developed by Idorsia, which is a spin-out of Actelion, which was acquired by J&J. And most of you probably indirectly know them through Data Warrior. So they originated Data Warrior at Actelion a long time ago. And I think scientists at Idorsia still maintain Data Warrior as a free service to everybody. So very nice of them. And finally, this molecule was a November 2020 molecule of the month. So very nice to see molecules that published great drug discovery stories actually make it across the finish line. Very cool. Second molecule is relatlimab. It is a LAG-3 blocking antibody. What's interesting about this is it is the first new checkpoint inhibitor immunotherapy mechanism approved since CTLA-4 and PD-1 and PD-L1 almost 10 years ago. So if you've been working in oncology, immunotherapy has been this crazy hot space, and there's been tons of investigation into what's going to be the next PD-1, right? Or at least what's going to combo with it. There have been targets like TIGIT, TIM3, OX40, CD73, STING, pursued by large and small molecules. And people, until very recently assumed that TIGIT was going to be the next target that, would show some efficacy when drugged. But Roche just announced a negative phase 3 trial in the first quarter. And so ironically, a molecule targeting a protein called LAG is going to be the winner in this race. One fascinating thing about the pharmacology of this is the half-life of the LAG-3 antibody. So this is a 2 antibody combination. They're administered together, a LAG-3 antibody and nivolumab. The half-life is 26.5 days, and it matches the other antibody's half-life of 26.5 days. So I don't know if they planned that or how they planned that if they did, but that is remarkable. And obviously, if you're giving 2 IVs, having those half-lives match and the pharmacology line up is really important. So would love to hear from somebody on that team how that happened. Third molecule, I'm not going to try to pronounce this one, but it is a PSMA targeted radiotherapeutic. Most of you are probably familiar with PSMA as prostate-specific membrane antigen. You probably can't open, endpoints or STAT without seeing some new thing targeting PSMA. It's a prostate cancer antigen, and it's widely used as a target for or a marker for antibodies, ADCs, bispecifics, CAR-Ts, etc. What I thought was cool about this was, I didn't realize that PSMA is also targetable by small molecules. It turns out it is also an enzyme called glutamate carboxypeptidase 2. Normally, when I think of these targets of biologics, I just assume that there's some big globular, basketball-shaped thing that small molecules bounce right off of. But it was interesting to see this target have a small molecule ligand. One tragic factoid about this area is PSMA was actually discovered by William Fair, who died of metastatic prostate cancer in almost 30 years ago. So it's a shame that he didn't live to see the fruits of his discoveries, but obviously it's a very important legacy to leave behind. Fourth molecule here is ganaxolone. It's a GABA receptor positive allosteric modulator. It's a neurosteroid or a steroid drug that is used for neurology. It's for treating seizures in a rare disease, CDD, and it has a 34-hour elimination half-life. So a couple interesting facts about this. So one is it binds to the steroid allosteric site on GABA. It's the same site that people believe is responsible for AR antagonist seizures. So if you follow the development stories of the drugs like enzalutamide, one of the issues there was those prostate cancer patients, a rare number of them would develop seizures as a side effect. And it was believed that the antagonism of GABA, because it also has a steroid binding site, and these molecules that target AR, are targeting a steroid pharmacophore. So it's interesting to see that. So you don't want to inhibit GABA because that can promote seizures. So if you're trying to come up with seizure medication, you want to agonize GABA to a certain extent. Another interesting thing about this is there was a recently approved drug, allopregnanolone, which is just an endogenous hormone. So Zulresso is given for postpartum depression, and it has a half-life of 9 hours and is given IV because it's not very bioavailable. One reason that, aside from not being bioavailable, one reason it can't be used for seizures is also the fact that because it's this endogenous hormone, it's also subject to these isoenzymes that can interconvert it to other species like pregnenolone sulfate, which are GABA negative modulators, which can cause seizures. So just adding a methyl group to that secondary alcohol here apparently makes it orally bioavailable, increases its half-life to 34 hours, and reduces the potential for these GABA inhibiting metabolites to form. Small change, but a big difference in drug properties. Mitapivat or Pyrukynd is pretty cool. It is a first-in-class allosteric activator of pyruvate kinase. When I saw kinase activator, my eyebrows raised a little bit, and I was like, "That's interesting." It is a very interesting mechanism. So technically, it's actually, even though it's structurally like a kinase, it has a hinge region and alpha helix, CNN terminal domains, etc. It's one of these ancient enzymes that has other functions. Before I guess proteins started getting phosphorylated. So what it does is it mediates the last step of glycolysis. It takes this phosphatidylpyruvate down to pyruvate and converts ADP to ATP instead. Kinases, by definition, use ATP and phosphorylate a substrate. This irreversibly does the opposite. So that's kind of cool. So therapeutically, the reason this is important is mature red blood cells lack mitochondria, and they depend on glycolysis to operate. So obviously, if you have a genetic disease where you can't efficiently undergo glycolysis, you can have anemia as a problem. So this molecule increases the activity of both wild type and mutant PK enzymes in patient red blood cells. And the important thing about this was they found that this was active against a broad range of mutants that cause this disease. So this rare disease PK deficiency is actually caused by hundreds of submutations. So it was really important that this drug that was already targeting a small population, could work against a range of those mutations. There's another PKA or pyruvate kinase activator in development. I want to say it's in phase 3 now from Forma, and that was highlighted in our last molecules of the month as well. So if you want to read more about this, there are 2 places you can find more information. Can't go on without talking about JAK inhibitors. Now, there's been so much written about JAK1 and JAK2 that I won't go into a ton of detail about these and how they differentiate. The main thing I wanted to highlight that I thought was interesting was JAK2's role in this whole story. So, the first inhibitors for JAK that were approved were PAN inhibitors. They inhibited JAK1, 2, 3 to a certain extent, and 4. And JAK2 was thought to be, cause of the side effect anemia because JAK2 plays a role in EPO and TPO signaling. EPO is erythropoietin. It's the doping drug that athletes use to get an extra boost by increasing their red blood cell production. So obviously, if you're inhibiting signaling through that pathway, that's not going to be good for red blood cell counts. So for second generation, third generation drugs, people tried to pursue JAK1 selectivity for inflammatory conditions and things like that, where they don't want to see anemia. But paradoxically, JAK2 inhibitors were actually found to improve anemia in a certain type of cancer patients, so myelofibrosis patients. And after doing some studies, they found that this was likely due to basically killing the pathological blood cell precursors in this cancer-like disease before it impacted the healthy stem cells. So it was a surprising finding. People didn't really expect it. And so people have now followed up with more selective JAK2 inhibitors to try and improve the therapeutic index because JAK1 comes with its own separate on target, pharmacology. And this drug on the right, pacritinib, showed clinical benefit, interestingly, in a subset of myelofibrosis patients who already suffer from thrombocytopenia. So previous JAK2 inhibitors like fedratinib were specifically contraindicated in that subgroup of patients because they were concerned that it might make the thrombocytopenia worse or more severe. But this drug, even though it has apparently limited selectivity in cells, only about 5x, it is enough to see efficacy here. So that's how it's differentiating. On the bottom right, there is another JAK2 inhibitor that recently read out that was acquired by GSK for $1.7 billion from Sierra Oncology. Anybody who's ever worked on a kinase program is probably looking at that molecule and grimacing a little bit, like, how is that selective? How is that a drug? But this all just goes to show, as much as we'd like our molecules to be beautiful, perfectly selective, and all that, at the end of the day, what patients care about is that it works in some disease that didn't have a treatment before. So efficacy always wins over elegance. So we didn't cover all the approvals, but hopefully that gave you, enough of a sense of what was going on in the first quarter, some interesting tidbits. Now, we did highlight about 30 molecules of the month over 3 months. So again, can't talk about all of them. I'll just pick out a couple that were mostly selected for diversity. Obviously, when we choose them for molecules of the month, we think they're all interesting, should continue to read about them. But we just wanted to pick stories that were different. So the first one that was cool was this Shionogi COVID main protease inhibitor. So everybody knows about Paxlovid at this point. It was our molecule of the year last year. That is a reversible covalent nitrile-based inhibitor of the main protease. One of the issues for that drug is that it needs a PK booster. So it's actually a combination of two drugs, nirmatrelvir and ritonavir. And ritonavir is also a protease inhibitor, but it's actually mainly given for its CYP inhibiting activities. So this drug apparently does not, has good PK on its own. It's oral, long half-life. And because of that, it has potential for fewer DDIs in COVID patients. This is going to be important because obviously the greatest morbidities and mortalities are in COVID patients who already suffer from, conditions where they are likely going to need other drugs. So diabetic patients, cancer patients, immunocompromised people. So even though, Paxlovid is being manufactured, I've heard from some suppliers that there's a scramble to acquire, 200 tons of certain intermediates, and there's this crazy race in China right now for being the key Pfizer supplier, I guess. But there's going to be room, I think, for additional COVID drugs, and this is an interesting one. The discovery strategy was also pretty cool. It's from a PharmacoPhore guided virtual screen. They confirmed about 300 hits by high throughput mass spec. And this is a good case study because, for virtual screening, people, like to talk about, oh, how big is your virtual screen? But if you've ever done a virtual screen, the hardest part about a virtual screen is narrowing it down to what you're actually going to test. Because it doesn't matter if you screen, a trillion compounds or whatever, you're going to get hits. And you can't test, even a fraction of a subset of them. So it was interesting to see them prioritize it and get hits from only 300 molecules that were synthesized. Next molecule is an older molecule, but it was just published. It's a partial GK activator, another kinase activator. I didn't plan for that. That's actually interesting. So GK activators were super hot in the 2000s. I want to say it kicked off in 2003 with this Nature paper on, characterizing the target. Basically, people found that GK is kind of this master switch for, setting glucose levels in the blood. And, when people read that paper, they were like, oh, that has obvious implications for diabetes. Now, the problem was, there were hundreds of patents published, tons of GK activators went into the clinic. I want to say there were like 20 clinical candidates at some point. But the main problem was on-target hypoglycemia. So you can activate GK, but then the problem is it goes too far and it reduces blood sugar levels to dangerous levels. And so BMS had a strategy of saying, okay, well, maybe what we want is a partial activator. So, this is taking that mechanism to the next level, right? So kinase activators are already pretty cool, and they're going to look for a partial kinase activator. And it seems like they were successful, and they took this molecule into the clinic. They had a phase 1 done in 2011 and 2012, and it was well tolerated and with some window against hypoglycemia. But the problem was the whole target class was basically halted because with other GKAs, people saw that even if you had efficacy, after a couple of weeks, the efficacy would disappear. So it doesn't matter if you had a safer drug, doesn't seem like it worked. What I thought was also cool, and this is because I'm not a nucleotide chemist, is this molecule is not a prodrug. So even though it has this phosphonate ester, with these two ethyl groups, it actually binds with those with that phosphonate ester intact. You can even see it in the crystal structure there at the bottom right, and the PD correlates with the parent molecule. After digging into the literature some more, I guess this isn't surprising in hindsight because nucleotide chemists spent decades trying to actually optimize phosphonate prodrugs until they came up with, protide technology that's used in a lot of nucleoside drugs now. But it was an eye-opener for sure. And the reviewer, Kim Hewer, pointed out a really nice section of their discovery story, where on the bottom left, that pyrazine heterocycle, they did really nice conformational analysis to see, which was the ideal heterocycle to enforce the conformation that they wanted. They looked at torsional effects and dipole-dipole interactions between both nitrogens and both side groups. So very interesting read. Next molecule is $1 billion molecule. So Sanofi bought Principia a while ago for $3.7 billion to acquire these reversible covalent BTK inhibitors. In this JMed Chem, they published a reversible covalent oral molecule as well as a topical molecule. So I thought this was interesting. because, obviously structurally a bit related to ibrutinib, and there's been tons of BTK inhibitors in development. What's nice is this is, to my knowledge, the farthest in a reversible covalent acrylamide has gone in the clinic. And the nice thing about this is now we have a phase 3 purely reversible molecule that's a fenebrutinib from Genentech in MS. We have this reversible covalent molecule, and we have several covalent molecules. So the whole theory behind reversible covalence is that, because they don't leave a neoantigen or neoepitope once they, once they finish inhibiting their target, the idea is that potentially these kinds of covalent drugs would be safer long term. So now we are actually going to be able to watch clinically, hopefully, as long as these molecules stay in development, we'll be able to see if that actually plays out with an N of 1. All right. The next molecule is INE-963. It's an anti-malarial. This was nominated by, so this was developed at Novartis Emeryville, and it was nominated by Dennis Koester, who does work at Novartis Emeryville, but we'll forgive him for nominating his colleague's molecule because it is actually pretty cool. This molecule is a super fast killer. It reduces parasite burden within 24 hours. And this is important for both symptoms and addressing resistance. You don't want to give the bugs time to come up with something new, I guess, or at least select for something. And so far, they've done a lot of selection studies for resistance, and they haven't found any resistance mutants through all the standard studies. But it does have activity against drug-resistant strains and field isolates. So it doesn't, the mechanism is not known, but, the fact that it has this, better activity against all these resistant strains suggests that it's some kind of first-in-class mechanism. It does have a long half-life, and, it has a potential to be a single-dose cure. And that's also important for resistance because, I don't know if any of you have seen this documentary about, malaria in the developing world, but because our artemisinin can sometimes be in such short supply, people, don't take their full courses, or they try to save pills to use just in case, or they break these pills in half, thinking that, if they just take half of them at a time, it'll extend the supply. Obviously that contributes to the development of resistance. So if you have something that can just be given in a single tablet and, that's what you take and it works, that would be great. Obviously, we'll have to see if this can be manufactured economically for everybody, but that does look very promising. Great. Now for something different. This is another kinase inhibitor, but it is a phase 2 candidate for neuropathic pain. I thought this was wild because, traditionally pain has had, even neuropathic pain has had a very high bar for safety. And traditionally, the fear with kinase inhibitors has always been selectivity and safety issues. And so, kinase inhibitors, have always tended to start in oncology where, safety is, it's less forgiving because the disease is so severe. But to go, from oncology to pain is quite a leap, I think, for a kinase inhibitor. And that is very impressive. Maybe it says that we're in an era now where kinases, could be considered, something like GPCRs, where maybe it doesn't matter what indication you go in if you can find the right selectivity. There have been, Jake Schwartz, one of our neurologists, pointed out that there are other CNS penetrant kinase inhibitors that have been in development, LRRK2, DLK, RIPK1. But these have typically been for more serious conditions like Parkinson's and these, so far as I know, I believe all 3 of these have had potentially on-target toxicities observed, although I think LRRK2 and DLK, I think people still have molecules that are in development for Parkinson's. Don't quote me on that, I have to double check. Point stands that this does appear to be an outlier in kinase development and should be a molecule to watch. One other interesting thing about the story is it does seem to be a molecule that was, a target, AC1, that was discovered in-house at BMS. They did this, crazy phenotypic screen of 3,000 or so mice strains looking for, genes that would have an impact on pain sensitivity. And they discovered that if you had one allele of AC1 missing, that desensitized these mice to pain. So maybe that's why they're keeping this target under wraps. Okay, branaplam is a splicing modulator. This is also an older molecule. It was previously a clinical candidate for spinal muscular atrophy, or SMA. Risdiplam is a drug that acts by the same mechanism that is approved. And what's been interesting about these splicing modulators is, number one, I still think it's crazy that small molecules can, bind to these, RNA protein complexes and change the way proteins or RNA is spliced. That is just mind blowing. But what's interesting is, one of the problems in the development of those drugs was they had to worry about splicing selectivity. So they target, RNA that binds to this splicing complex, but that's involved in the splicing of a lot of different genes. And I think they narrowed it down for risdiplam to it only affects 20 other splicing events or something like that out of the universe of thousands of potential interactions. But as people dug into these molecules further, they found that some of these off-target splicing events are actually pretty interesting. One of them being Huntington, which is the main driver of Huntington's disease. If you've watched Grey's Anatomy, this is a disease that doesn't have a lot of treatment options, typically fatal and progresses. So if you have the genes for it, it's almost inevitable that you'll get it and die from it. And this mutant Huntington protein is basically a prion that aggregates. So the thinking here is maybe if you have some of these splicing modulators that can reduce HTT expression, maybe that would have an impact for the first time in this disease. So that's pretty cool. Another company, PTC Therapeutics, which also helped originate risdiplam in collaboration with Roche. They have a CNS penetrant phase 2 candidate with this mechanism in Huntington's disease. They haven't disclosed their structure, but I'm sure both of these molecules will be, hotly watched. We still don't know whether the safety of lowering wild type HTT is going to be an issue. But the cool thing about this is because branaplam was in clinical trials for SMA already, they were able to see that HTT is modulated in those SMA patients. And so far, those patients have been fine. So it seems like a very good, therapeutic rationale. But we'll have to see. I mean, since this disease does take a long time to manifest, these may be long trials similar to Alzheimer's, but very cool stuff. All right, finally, we'll wrap up with a couple conference disclosures. So there were 6 candidates disclosed at ACS and at least 12 candidates disclosed at AACR, probably more than that. We've got 10 of those highlighted on our site, and people keep submitting new structures, which is great. Really appreciate it. We'll get those new disclosures up soon. But here are just a couple ones that I thought really stood out in terms of novelty or impact. So AMG 650 is super cool, targets KIF18A, which is a motor protein. Motor proteins are not like GPCRs, they're not things that we typically think about drugging, but there's been more activity in them in recent years because Cytokinetics, Myocardia, and other companies, and Amgen has actually been active in that space as well, had found that myosin can actually be modulated both by activators and inhibitors for different cardiovascular diseases. So this is cool because they're in oncology, but they're also targeting a kinesin motor protein. And they took a pretty rational approach. If you look at their JMedChem paper, started with a screen, found some good chemical matter, further characterized it, really found that it actually bound to the target and developed a candidate. And so maybe this suggests that motor proteins as a class are more druggable than we appreciate. And I'm sure that there will be more targets soon in this space. I think BMS signed a deal with another company on this area of chromosomally unstable cancers. And that company starts with a V. I can't remember what it was, but they also have a KIF18A program. And so I suspect that this will be an area to watch. KVD900 from KalVista is a plasma kallikrein inhibitor. So this is going to be second after berotralstat if it is successful, but it is differentiating based on fast action. So a little bit of context here. HAE is this genetic disease where people you lose this gene, C1 inhibitor, which is in the complement cascade, and basically the bottom line there is it leads to an increase in kallikrein activity, which increases bradykinin when people have these events, which is a super potent vasodilator. And they get this extreme swelling that's kind of like hives, I guess, but can be lethal. I thought about putting a picture on the slide, but just in case people get queasy, it's not the worst thing in the world. It's not like looking at neurosurgery or something like that, but you should Google acute HAE attacks and look at the images. It's pretty dramatic. These people are otherwise like totally healthy looking, but they get these HAE attacks and they just balloon because of this lack of one enzyme. So berotrostat, the previous molecule, structures on the top right. When it was approved, it was specifically for prophylaxis and limited in the label against use for treating acute attacks. What they wanted to do with this molecule was show that it could actually work as well in this acute phase and stop or reverse the attacks. But in order to demonstrate this, they had to come up with a faster assay because the assays people were using before to measure progress in this complement cascade took hours, and obviously this attack happens within minutes, and so you have to know if your drug is working quickly. With this new assay, they were able to find that they can activate their target within 20 minutes to 3 hours. And in the clinic, they did meet their phase 2 endpoints. They also met secondary endpoints, and I think they're using one of their secondary endpoints actually as their pivotal trial endpoint. And they say that they could be ready to file an NDA on this in 2024. So it will be interesting to watch how this space develops. Blueprint's Blue 945 is pretty cool. It is a 4th generation EGFR inhibitor. EGFR is a very old target. So the first drug was approved more than 20 years ago now, I want to say. But the fact that we're still seeing new drugs for it, kind of says how important it is and how many people, do have cancers that are driven in part by this pathway. Some of the key challenges with targeting EGFR, have always been selectivity versus wild type, resistance, and brain metastases. So people with lung cancer, and in particular people with lung cancers that have mutations in EGFR, they tend to progress by having brain metastases. So if you don't have a brain penetrant molecule, that could be problematic. I think it's something on the order of 30% of people actually develop these brain mets in these subsets of lung cancer. So it is a significant problem. So they are taking a best-in-class approach. They have 1000x selectivity against wild-type EGFR, intracranial activity in a patient-derived model, and apparently it has active against this triple mutant and some other mutants as well. And so Blueprint is pursuing, very aggressive EGFR combo strategy where they're taking, various potential best-in-class inhibitors and combining them to, have best-in-class combination. And those should be pretty exciting. They recently acquired a small company, Lengo Therapeutics, which was preclinical. They had a brain-penetrant EGFR exon 20 inhibitor. So EGFR exon 20 is another sort of hot target in this area. And I think they paid $250 million for this upfront for this preclinical asset. So it kind of says how important this combo strategy is for them. Another area to watch. Okay, finally from AACR, there were a lot of cool stories published. We have longer descriptions on our site, and we link to some very nice write-ups by the AACR team on these molecules in more detail. But I just wanted to share 3 mechanisms of action that I thought were super cool to see going into the clinic. First one is this PDE3A SLFN12 molecular glue, or what the authors call Velcrin. It came from a Broad Institute hit that they published in a 2015 Nature Chem Bio paper. And it's nice to see that, they've done SAR in collaboration with Bayer, and Bayer is taking a molecule into the clinic. There's a really cool mechanism. It helps induce this tetrameric complex. And for molecular glue, what's interesting about it is it actually stimulates SLFN12 RNase activity, which leads to cleaving of its substrate, this tRNA, which leads to ribosomal pausing and death of certain cancer cells. What's interesting about that is just that it's a gain of function glue mechanism. A lot of times we think of, first thing that comes to mind is these cyclophilin glues that Warp Drive and Revolution have been, advancing is, think about inhibiting the target that they bind to. This increases the activity of the complex molecule. I mean, I guess technically PROTACs or molecular glues that induce degradation are technically, well, I don't know, they increase proteasome activity, but they're reducing their target activity. Anyway, interesting new mechanism, check it out. Second molecule targets a phosphatase. So if you've seen a phosphatase high throughput screen readout before, you know that the things that come out are ugly. Like they have, phosphates, phosphonates with 2 negative charges or like multiple charges, things you typically don't want to work on. So it is impressive that AbbVie was able to find a clinical candidate from screening against the phosphatase and optimizing it. It is a picomolar inhibitor, and they confirmed the binding to their phosphatase target by X-ray. So that's pretty cool. A lot of times, in these phosphatase screens, you will see some, potent inhibitors, but they don't act by mechanisms that you want them to. We have an article on acid interference compounds, by the way, coming up soon, so stay tuned for that. But it is going into phase 1 in solid tumors, both in IO combinations and as a single agent. So very cool. Another difficult to drug target class that is finally being cracked. On the right, I have a soft spot for deubiquitinase enzyme inhibitors, especially allosteric DUB inhibitors. So it is cool to see a USP1 inhibitor going into the clinic also for cancer. The USPs, or deubiquitinating enzymes, they do the opposite of E3s and that pathway. Basically, instead of tagging proteins for degradation by ubiquitinating them, they mediate the opposite process by clipping these ubiquitins off, leading to stabilization of enzymes. So you can think of E3s and DUBs like kinases and phosphatases. They do the opposite. So another target class to watch. All right, well, I hope that this quick run through of all of these targets and compounds and developments were interesting to you. Again, I just pulled out some gems that I thought were particularly interesting, but if you want to read more, we do have deep dives, both for molecules of the month and approvals and all that. Basically, if you subscribe, you can get these slide decks where we basically do a full literature review and we put it all into one page so you can find information about the industry context, why people are pursuing these, what's the clinical status of competitors, things that authors will not tell you in their papers, along with, where the hits actually came from, past papers, pharmacology, mechanism of action, all that good stuff. You can read it all over 15 minutes over coffee and get a digest of all the cool stuff that's happened in the month. We do the same thing for approvals. We've just released a couple of our 2021 approval slide decks where we do the same thing, talk about the industry context, the history, why these drugs are important, what physicians are going to think of them, and what the key questions are for the future in these target classes. We are rolling out more and more new stuff every day. We are just starting industry reviews, including looking at mergers and acquisitions, the targets, lead mechanisms, competitive landscape of these molecules, and doing monthly updates now of new approvals. So each month we round up and we do the same deep dive for all of these new drugs. So if you're interested in, what is the competitive landscape? Why are these molecules special? What strategies are they used to differentiate? Consider getting a subscription for your team. Finally, we make all of this stuff searchable. So even though they are in these PDFs that you can share with your colleagues, you can also search on the site, for keywords like CNS penetrant or DEL screen and find these deep dive case studies. And so if you're thinking about, what have other people done to make a CNS penetrant molecule competitive or best in class, consider checking that out. One recent head of discovery at a large pharma company told us on a call that her subscription saves her days, if not weeks of time. So if you feel like you could use some more time for you or your team, get in touch. All right, so I hope that was helpful. Stay tuned for part 2. And thanks to everybody on our team for making this happen, especially Melissa and Spencer here for organizing our first webinar. And with that, I think we can go to questions. Melissa or Spencer, was there anything that popped up that you think we should start with?
Yes, so we'll start by the most recent. Did any of the hit drugs violate Lipinski's rule of five? Is one question.
Oh, I'm sure they do. Yeah, I haven't, done a tally myself, but I think if you look through the molecules of the month, we do try to, find some of these meteor compounds, I guess you could say. But that's definitely been an emerging trend. I think we're definitely seeing a lot more, beyond rule of five compounds. So, let's take a step back. If we're talking about hits, normally we don't think of rule of five. We think about, maybe fragments like rule of three or something like that. But, the lead molecules that, these go into, there have been some pretty impressive structures recently.
Hmm. Great. Well, thanks for that. Another question is, what about the use of JAK inhibitors in alopecia? Does selectivity towards JAK1 help there?
I would assume so. Alopecia is one of these, okay, hair loss, right? What side effects do you want if you're, if you're trying to treat hair loss? Probably none. So I think as selective as you can be, that would be great. Now, alopecia can have a significant impact on quality of life. And so maybe some people are willing to take bigger risks there. But in general, in these, immune mediated conditions where, people are otherwise fairly healthy, I think selectivity, is going to be important. I think that there are topical JAK inhibitors now, where the safety play there is basically making them tissue restricted, so making sure that they're cleared quickly and not absorbed as much. But yeah, it's an area to watch. I think the topical, tissue restricted inhibitors are going to be very interesting. So whether that's skin, gut, or lung, I think those are all places to watch.
Great, thanks. What is your view on the current state of cancer therapies? Do you think we're good at treating cancer despite all the money that's spent? Some feel efficacy is poor. Thoughts?
Yeah, that is a great question. So I think, one of the most exciting, okay, three super exciting areas to me. One is cancer immunotherapy, both with, CAR Ts, large molecules, and small molecules now. So with immunotherapies, like, we mentioned PD-1 and CTLA-4, the amazing thing about those is, they don't work for everybody, right? But, there are all these cases where patients on those checkpoint inhibitors, they can survive for 10 years or more, after being fourth line in metastatic melanoma, for example. I think that's only in like 13% of patients or something like that. But it's a significant number of people that effectively have a cure because of those therapies. So small and large molecule immunotherapies, I am super excited about. CAR Ts are very interesting. I had my blood drawn recently, and I was talking to a nurse who works at OHSU, and he works in the CAR T clinic, and he says basically all the time they are seeing these dramatic complete responses in patients with like lymphoma and things like that. So I was super skeptical of cell therapies, partly because of the cost and all this stuff. But it's true. I mean, they are opening treatment options for a small group of patients that are eligible, but are really leading to dramatic responses. So super exciting.
Awesome.
Yeah, I guess I shouldn't dwell on that. I mean, I could go on about the cancer treatment landscape forever. So I'll stop there.
Great. Okay. Any upcoming disclosures in AD and PD?
Atopic dermatitis. I mean, yeah, these are big areas. Yeah. So yes. I'll say stay tuned.
Stay tuned.
Stay tuned next summer.
Okay. Any interesting PROTAC molecular glue disclosures in recent conferences?
Yeah, so check out the AACR page. There were two interesting ones from C4 Therapeutics. Those were the sort of the big popular ones. Yeah, they have structures. That was the great thing about them. Although they are on targets that have been targeted before.
Great. Okay, I think this question has been answered. Here's another one. Biosys kallikrein inhibitor has a warhead benzylamine. Folks, I am not a scientist. The second in class molecule you show, it had no changed moiety. Is that a different target?
Firecryst's kallikrein inhibitor has a warhead benzylamine. The second-in-class molecule has no charge moiety. Oh, okay, I see. Well, it's interesting. It's the same target. They're both targeting plasma kallikrein. Now, let me double check on that if that's the same binding site. I'm not sure if, yeah, I'm not sure if they bind to the same site, but you're right. It is interesting that they're different because the first molecule, yeah, it has those two charged amines, which I want to say they have a crystal structure of. Yeah, let me look into that and get back to you.
Okay. Thoughts on recent approval for Alzheimer's disease?
We're talking about Aduhelm?
Sure, that was a vague
Oh,
question, but
yeah. Yeah, so we have an article that I've been thinking about publishing for a while. I know Jen and Naveed have been working together on it since the approval, and I've waffled a lot about whether we publish it or not, or how we publish it. You know, it's tough because it's obviously not doing what people want out of an Alzheimer's treatment. You know, the question is, right, did the FDA make the right decision? And a lot of people say no. All I'll say about it is, I guess, I think it's very interesting that we had an antibody that was brain penetrant, which is also another interesting story, right? How does this thing, you know, putatively modulate A beta in the brain? I think there's more interesting science there than people tend to talk about. And I'll leave it at that.
great, thanks. Any recent approvals started with FBDD hits?
Recent approvals from FBDD hits. I'm sure. KRAS, sotorasib, Amgen molecule. All those hits were from this tethering disulfide fragment screen from Kevin Chokrat's lab. That's probably the best recent example. We also have an article on FBDD coming up soon.
Great. Okay. Are there any NMDA antagonists in development for the treatment of seizures? Or the glutamine receptor antagonist or partial antagonists for the treatment of cognitive disorders?
Yeah, so that's one where I would say our search function is fantastic. I'm sure we have covered NMDA. Actually, that was one I remember talking to Jake Schwartz about recently. So NMDA is a very old target. Yes, I would search the site for more details.
Great, thanks. KRACS or PROTACs?
KRAS or PROTACs, both.
Porcano dose. Okay. Are you open to mentoring undergraduate researchers passionate about drug discovery?
Oh, that reminds me. Yes. You know, we typically work with graduate level students, but feel free to get in touch. I forgot to mention that we are hiring actually, but this is more at the more senior level. So if you know anybody who is interested in being something like an associate editor or somebody you trust to write about science for you, please put them in touch.
Great, thanks. Let me see if we have any more questions. Oh, how can molecules like ganaxolone get its IP with only one methyl difference?
Yeah, so IP is something we could have a whole seminar on. There's a couple strategies you can use. So if you're asking about composition of matter, the important thing is that you can demonstrate that this was a non-obvious change, and that it has a significant consequence, I guess. So yeah, I mean, I would say that was not the magnitude of that effect was not obvious to me, but again, don't quote me on that. I'm going to get dragged into a courtroom or something now. But I'm sure they've also got method of use patents and other forms of IP that are not composition of matter. Certainly, I think Zulresso, that one, which is the endogenous hormone, that one, I'm pretty sure is just protected based on method of use and formulation.
Excellent. I'm looking to see if we have any final questions. I believe you have answered all that have been submitted through the Q&A box as well as the chat. Does anyone else have any questions?
Cool. Well, thank you all so much for coming. I hope to see most of you at part 2, for those of you who are interested to learn more about what's going on in the industry. And yeah, thanks so much for coming, and stay in touch.
We do have one more.
Oh, okay. All right.
One final question. Can we expect a chemistry with drug discover perspective?
We'll have more chemistry articles coming soon. Yeah, definitely stay tuned.
All right. Thanks, everybody.